Hello, everyone. I'm Pete Rand. I'm with the Prince William Sound Science Center in Cordova, Alaska, and it's a great pleasure to be able to present to your group, Sapporo Wild Salmon Project. Really appreciate all your efforts to restore wild runs of salmon to the Toyohira in Sapporo, and I hope to be able to support any efforts that you have moving forward. Your work is very important, and it's a great pleasure for me to present some of the work that I'm doing, and hopefully it'll be beneficial to your group. So I'm going to go ahead and share the screen. Okay, so yeah, I'm Pete Rand. I'm a research scientist at the Prince William Sound Science Center, a small conservation educational group based in coastal Alaska in the United States. And I also volunteer as a specialist group chair for the IUCN, the Species Survival Commission, based in Switzerland. And the focus there is on protecting fishes in the salmon family throughout the world. The title of the presentation today is Biology and Status of Wild Salmon in the North Pacific. And this is a very broad subject, of course, and I only have about 40 minutes to present. So I'm going to be touching on a lot of different topics briefly, but if you have any questions, I'll be happy to answer them. So this is a recording, but I'm hoping to be available virtually on the day of this event, and if any, and hopefully I'll be able to answer any of your questions. Before I begin, it's important to acknowledge native lands. Everywhere I work and live in the North Pacific working on salmon, I'm on native land, and I recognize, appreciate, and honor Indigenous peoples. We are committed to building long-term relationships with Indigenous organizations and elders to create relevant and actionable information in support of their sovereignty and self-determination. I took this picture at the headwaters of the Copper River in Mentasta Village. Salmon are very important cultural resources for this small village and many villages throughout the Copper River watershed where I work. And it's important for me to acknowledge this. This is just a brief outline of the presentation today. I'm going to describe where I work in Alaska. I'll give a biological description of Pacific salmon, fishery, salmon, global markets, threats, and the status of Pacific salmon. I'm going to highlight some of the research in Alaska, focusing on salmon migration. I'll touch on some of the research I've been involved in over a decade now in Hokkaido, and then hopefully have some questions, time for questions at the end of the presentation. I grew up fishing and I love the outdoors. I'm fascinated with the biology of salmon, and I've devoted my whole career to studying them. This was a picture taken of me during my first trip to Cordova, and I was fishing for coho salmon. Fishing is something I really enjoy doing, fly fishing, and I take every opportunity I can get to fish for salmon in Alaska. I live part of the year in Cordova, Alaska. This is a very small community, about 2,000 people. It's located in the eastern part of Prince William Sound in south central Alaska. Seafood harvested by the fleet that you see here includes Pacific salmon, Pacific cod, Pacific halibut, sablefish, crab, and shrimp. And currently, there's a lot of interest in developing a kelp industry here. And we certainly look toward Hokkaido as a model. And I'm expecting to see a lot more growth in the kelp industry in coastal Alaska in the future. Our Research and Education Center was created after this important historical event, the Exxon Valdez oil spill that occurred in 1989. Over 11 million gallons of oil was spilled into Prince William Sound and had a dramatic effect on marine life and the seafood industry in this region. I work with a variety of colleagues to understand the long-term impacts of oil spills like this on marine environments. There are now many more precautions in place to prevent oil spills in the future, but there's still a risk of these oil spills occurring again. I focused work on Pacific salmon in 3 broad regions: the state of Alaska, the Russian Far East, and Japan, shown here. After presenting some of the background information on Pacific salmon, I will describe an ongoing project I lead in Alaska, and then talk briefly about a project I'm still involved with in Japan. This is a map that many of you may have seen before, but it shows the global distribution of Pacific salmon. And you'll see the green shaded areas here are salmon watersheds. This represents the freshwater habitat available to Pacific salmon. And you see it extends from the Gulf of Alaska and the Sea of Japan up and includes Sea of Okhotsk, Bering Sea, and also increasingly the Arctic Ocean. As the globe warms, we're finding more and more salmon distributing into the Arctic Ocean. So this is an important recent phenomenon. And then the dark blue indicates the waters occupied by salmon in the North Pacific and the Arctic Ocean. To understand the status of salmon, you need to understand how humans are affecting them. This is an image of the density of humans in the North Pacific. Human civilization has depended on salmon for thousands of years, but our level of effect on them and the rate of exploitation on them has increased dramatically in the last century. And as you can see from the distribution of humans, the impact is disproportionately high in the southern part of their range, whereas in the north, the watersheds and the habitat are in more pristine condition. This is a simplified description of the life history of salmon. The key life history trait these fish is anadromy, which means they are born in fresh water, migrate as juveniles to the ocean, and return as adults to fresh water to close the life cycle. It's important to acknowledge this is an oversimplification of the life history. There are many exceptions to these patterns, and there are many biologists actively studying unusual life histories in some of these populations. This includes a lot of my Japanese colleagues, which are doing excellent work on understanding salmon life histories. It's also important to note the importance of the nutrient cycle here, which is often overlooked. So it's shown here in this diagram with a decomposing salmon carcass. But after these fish spawn and die, they release energy and nutrient into the aquatic food web. And this is really important for maintaining productivity in a lot of our streams. This is a simple illustration of the fates of Copper River salmon during their salmon migration. So this just gives you a better understanding of what happens to these fish once they arrive back into their home river. This is I'm showing a pair of adult sockeye salmon returning. Many of the fish are captured in coastal fisheries. This is a gill net boat shown in the estuary in the Copper River. Once they ascend up in the river, they're vulnerable to capture by a number of other fisheries, including upriver subsistence fisheries, sport fishermen, and personal use fisheries. Copper River has an active sockeye salmon hatchery, so some of these fish are taken as brood stock. Eggs are taken and fertilized and reared over the winter, incubated over the winter, and then they hatch in the spring and they're released into the river the following year. And of course, some of the fish are able to reach the spawning grounds and spawn in natural stream environments as well. This is a time series of the commercial catch in millions of salmon across the North Pacific from 1925 to more recent years. And the colors of the bars in each of these years indicate the composition of species. So we have pink chum, sockeye, coho, chinook, cherry salmon, and steelhead. It's important to know that there are many more salmon in the ocean now than there have been since these records began. And it's also important to know that 40% of the total biomass now is currently salmon that originated in hatcheries. So a significant contribution of hatchery salmon across the North Pacific. I will describe some recent results of what kind of effect these hatchery salmon are having on wild salmon. Here's a depiction of the kind of primary sources and the fates of salmon. Pig salmon are shown in this diagram, but as I mentioned, there's certainly a lot of wild production, but there's also a lot of hatchery production across the North Pacific. Many of these fish come back, mature, and are able to reproduce in natural streams, but as a large percentage of them are caught in the fishery or serve as broodstock for hatcheries. And this is an example of a canned pink salmon, which is a major commodity produced in Alaska. And these cans and other seafood products enter a global market. So these fish can be exported to many countries across the world. So they're a really important economic driver for Alaska and for other regions in the North Pacific. So it's important to acknowledge that. It's also important to recognize other fisheries, including subsistence fisheries, which has seen declines in recent years. It's become a big concern in Alaska, and important sport fisheries as well. As I mentioned, I volunteer with the IUCN. One of the most important products of the IUCN is the Red List of Endangered Species, which compiles conservation status of over 150,000 species across the world. I've been focusing on assessing the status of fishes in the salmon family, known as salmonids. This is one example of our work, a range-wide status assessment of sockeye salmon. Each subpopulation in this assessment is color-coded based on status. It's described in the legend in the lower left-hand side of the figure. The basic pattern we see is relatively healthy, stable populations in the north, but threatened and endangered populations in the south. And this is a pattern that we see in many of the other species as well. And the reason for the decline in salmon, I'm going to be covering in upcoming slides. NOAA is short for National Oceanic and Atmospheric Administration, and it's the agency responsible for assessing the status of Pacific salmon in the United States. So this table, which is, I apologize, it's hard to see, but each row basically represents the status of what's known as evolutionarily significant unit, or ESU. And these are groups of related salmon that represent a unique population that have unique traits. There's a total of 52 of these ESUs throughout the US Pacific Northwest, and nearly half of them are currently considered threatened under the Endangered Species Act. And unfortunately, there's little evidence that the status is improving. This is a dated assessment from 2016, and I understand that NOAA is going to produce an updated assessment later in this year in 2023. I want to just describe some of the key threats and the need for conservation. As many of you know, habitat loss and degradation is one of the key drivers here. A lot of natural habitat has been degraded or completely lost. And as I've mentioned, a lot of this has occurred in the southern part of the range, but it is happening to some degree in nearly all parts of the Pacific salmon range. Dams and other barriers to migration are also an important impact. There's a great trend that we're seeing in the US of removing dams and allowing passage of fish by dams. That's opening up historic habitat. So that's a trend that's happening and is continuing in the US, and we're seeing that in other regions as well. Overfishing is also a concern. Hatchery impacts, which I've noted already. And then, of course, the big one is climate change. And so I spend a lot of time thinking about this, and a lot of the research that I'm focusing on has basically an objective of trying to understand how climate change is going to impact salmon in the future. Here's an example of habitat loss. This is a picture I took in Hokkaido, modified stream channel running through an agricultural field. There are many examples of loss of natural habitat throughout the range of Pacific salmon. And again, a lot of this has disproportionately impacted the southern part of the range for many of the species. There's illegal fishing. It's referred to as illegal, underreported, and unregulated fishing, or IUU fishing. And in the Russian Far East, which is where these pictures were taken, it's meeting a demand for caviar, ikura, or salmon eggs. And this picture was taken of an illegal gillnet fishery in Sakhalin, Russia. Eggs are harvested from female salmon, salted in bags, and sold in domestic markets in Russia. Unfortunately, much of the resource is wasted. This is a picture of fish that have been basically slit, the eggs taken, and then the bodies left on the riverbanks to decompose. So it's a lot of wasted resources. And this illegal practice is relatively common. I've seen it firsthand, some of my trips to the Russian Far East, and it's very hard to control. Salmon hatcheries have been developed for over 100 years across the North Pacific. There are a variety of different types of hatcheries with different objectives. Broadly, there's ones that are focused on augmenting production of wild runs, and these are referred to as production hatcheries, and others focused on recovering threatened runs. These are commonly called conservation hatcheries. Over 5 billion juvenile salmon are released each year, and each dot, the red dot presented in this map, represents a hatchery. You can see that there's many hatcheries in Japan, but also we have a lot of hatcheries in Pacific Northwest and southern British Columbia. In terms of volume, Japan and Alaska are the lead producers, and that's mostly pink and chum salmon hatchery production. And again, these hatcheries do have some limited benefits to supporting fisheries and helping to restore threatened and endangered runs, but it's important to acknowledge that there are a number of risks associated with them, including negative interactions arising from disease, competition, mostly in the open ocean, and also predation. This is an example of a field project I am leading in Alaska. This is a collaborative study involving a state resource agency, the Alaska Department of Fish and Game, the seafood and hatchery industry in Alaska, NGOs, and universities. The project is called the Alaska Hatchery Research Program, and it started in 2012, and it's still operating. The project involved intense field work for field crew in Prince William Sound over 8 years, and we processed over 300,000 adult pink salmon. Study was possible because we were able to distinguish the origin of all the fish that we sampled, all of the adult salmon that we sampled, by identifying thermal marks in the otoliths, which are also called ear stones made out of calcium carbonate and located in the brain cavity of these fish. The picture on the top left-hand side shows one of our field crew extracting an otolith from the brain case with a set of forceps. So we were able to identify origin of all of the fish that we sampled in the field, and then we were able to sample the offspring produced by those spawning parents in future years. And so by sampling the adults and identifying the genetic markers, we're able to assign all of the offspring back to the parents that produced them. And because we knew the origin of these fish, whether they were hatchery or wild, we could compare the relative reproductive success of each of the pairings that we identify through our field and laboratory work. And so then we could understand how successful hatchery salmon are at producing offspring compared to their wild counterparts. And these are some of the results from the Alaska Hatchery Research Project. And this was from a paper in Evolutionary Applications that was published last year. Kyle Shedd at the Alaska Department of Fish and Game was the lead author. And I'm showing results from Stockdale Creek here, but there were results from two streams. Very similar results were found in both streams, but I'm only showing results from Stockdale Creek here. The x-axis here is the proportion of parents in an odd and an even year, and then the number of offspring produced from those parents, ranging from 0 up to 20. And the origin of the fish are identified by color, as you can see in the legend. So the key result from this is that if a hatchery origin salmon is involved in spawning, a reproductive event in a natural stream, their success at producing offspring was reduced by about a factor of 50%, ranging from 42 to 60% compared to wild salmon. So these results suggest salmon that stray from hatcheries and enter these natural streams could have a negative effect on overall production of wild salmon. So this is really important results. It has relevance to certification through the Marine Stewardship Council. And there's been basically a lot of talk recently on some possible changes in the way that hatcheries are managed in this part of in Prince William Sound. So there's going to be continued discussion about what this means in terms of managing salmon in the coming years. As many of you probably aware, we're experiencing a greater frequency of heat waves in the North Pacific. This is a map of these recent heat waves in 2015, highlighting 2015 and 2016 in the North Pacific. And it's also been referred to as the blob. But this is basically representing sea surface temperatures, but it's important to note that this effect was seen in deeper waters as well. And this phenomenon was associated with many negative effects on marine life, including die-offs of seabirds. And another heat wave occurred in 2019 that I'm going to touch on in an upcoming slide. But it's important to recognize that these heat waves are having negative effects on a lot of different marine life, including salmon. At the same time, the oceans are warming. They're also becoming more acidic due to increases in carbon dioxide in our atmosphere. There's been some studies that have shown potential negative effects of increased acidity on shell-forming organisms in the marine food web. These are images of pteropods, also called sea snails, which are an important source of food for juvenile salmon in the ocean. But they're finding it more difficult to form and maintain their shells in an ocean that's becoming more acidic. And these researchers have at PMEL have predicted that this is going to be significant by the year 2100. And this could have dramatic effects on the marine food web. So not only are the oceans warming, but there's changes occurring in salmon prey that could have important implications on the food web. Many people think about global climate change and think about droughts and heat that might be affecting salmon, but we're also seeing different kinds of effects in regions that are heavily glaciated. So this is an example of the top right here is showing the loss in mass of glaciers in South Central and Southeast Alaska. And the dark colors and the reds and oranges indicate areas that are losing a significant amount of glacial mass through glacial melting. The figure to the left here shows discharge increasing in the Copper River, and this is primarily driven by accelerated glacial melting. And I should note in 2019, circled here in red, this was that unusually high air temperature year in Alaska, and that led to unprecedented amounts of glacial melting and flood conditions in the lower Copper. So this is going to have a cascading effect on a lot of different organisms, including salmon in the Copper, It's going to increase scouring in some of the river habitats that could affect eggs incubating in gravel. It's also going to make it more difficult for adults to ascend up the rivers and reach spawning grounds. And so these are the kind of things that we're focusing on in our research in Alaska. While all of this is going on, we're seeing fish coming back at smaller sizes, smaller body sizes, and it's particularly dramatic for the Copper River. I'm showing some results of, this is size at age, data from the Copper River fishery, indicating that by, over the past decade, there's been significant reductions in body size of fish captured in the fishery, been particularly dramatic in recent years. And so this has raised a lot of questions about their fitness, their ability to successfully reach the spawning grounds and close the life cycle. So this is an important thing that we're looking at. I want to spend a few minutes now just describing a project I've been leading in the state of Alaska. And this is, I also want to acknowledge my primary collaborator, Dr. Kristen Gorman, who's a faculty member at the University of Alaska Fairbanks College of Fisheries and Ocean Sciences. We began this project back in 2016. We got significant funding to start the project in 2019. And so for the past 3 years, we've been collecting a lot of data. And this is a kind of a big multi-collaborative project involving a lot of people. I don't have time to fully describe all of this, but it's important to recognize kind of a main questions and some of the factors we're looking at and some of the primary methods. So this slide introduces that. Our prime question is what factors might be related to mortality risk for migrating salmon while en route to the spawning grounds. So as I mentioned, with increased glacial melting, we're seeing higher flows. And in some tributaries, we're seeing warmer water temperatures. And so we want to get a better understanding of how these changes in river conditions might be affecting the risk of mortality of adults during the spawning migration. We're looking at a number of different factors, this declining trend in size, as I mentioned. There's also indications of declining condition or the amount of fat the fish are coming back to the rivers with. We're looking at different spawning populations or spawning stocks. Some of them enter tributaries with a lot of glaciers, and others spawn in streams where there's very little glacial input. So we're trying to understand how each of these different populations are responding. We're looking at disease, pathogen prevalence in different populations. And we're also taking genomic approaches to understand how genes might be responding to these different stress factors. There's two primary field methods we're using. We're basically collecting fish and looking at body composition, specifically size, shape, and fat content, and also looking at evidence of gene regulation, documenting gene regulation in these individual fish. And we're also collecting fish, tagging them, and tracking them upriver. This is the radiotelemetry component of the field project. So again, this is kind of a broad, relatively complex project. I'm going to be focusing here just on the radiotelemetry work. And we've been able to summarize some of the results, and I'm going to present that to you today. This is just an overall description of one of our project components, which is looking at body composition. So we're collecting fish in the lower river. We're also collecting fish near the spawning grounds. These red fish you see at the top right are adult sockeye salmon collected near a spawning area in the upper Copper River. Male is on the top, female on the bottom. We're making measurements in the field. We're bringing these fish back to the lab, and we're doing a full workup on size, condition, and health metrics for all of these fish. And we're doing this for the different populations and the different run timing components, early, mid, and late run fish. So over the course of 3 years, we had a radio telemetry project. We were able to tag over 800 adult fish over the 3-year period. This is a picture of a fish in the flow-through trough that we were able to make all of our measurements and tag the fish just prior to release. We collected a lot of the fish with fish wheels with help from the native village of Eak. And in some cases, we dip netted fish from the bank of the river. This is one of the most amazing field sites I've been able to work in Alaska. And the crew is shown here dip netting for salmon. And on the other bank of the river is Child's Glacier. And we worked here for several days in July of 2020. And almost every hour we saw that calving, the glaciers calving, and then on 3 separate occasions, a wave caused by the water displaced by the calving traveled over to the bank that we were working on. So it made for very exciting field conditions. But it's just an incredible river to work in, and just really enjoy the field work. Just some details on the tagging component of the project. So once we capture the fish, either by dip netting or on the fish wheel, we place them in these troughs, and we collected a lot of biological data on the shape of the fish and the size of the fish. And we again focused on different run timing groups early, starting in May, mid-season, and then in August was considered the late run fish. An objective here was to understand what body characteristics are related to successful migration. And this is the fat meter. I'll refer to this in upcoming slides as well. This is a handheld device that you could put up on the side of the fish and basically press a button, and it gives you an instantaneous measure of the fat content of the fish. So it's very useful for our work, and it doesn't affect the fish at all. And you can very quickly determine the level of fat. So it typically goes from low values, about 1% or less, to 10% or sometimes greater. And we definitely see a pattern of early run fish with higher fat content, and then fish arriving later have much lower fat content. Once the fish are tagged, we collect all the information on the fish tag and release them. Then we track them through the watershed. So this is a map of the watershed, and we worked with the Alaska Department of Fish and Game to deploy and maintain radio receiver stations. So each red point here on the map is a radio and a antenna and a radio receiver. So when the fish pass by each one of these receivers, they're detected, and each tag, of course, releases a unique code, so we know exactly what fish is being detected, and all of that is stored digitally in the receiver. And so over the course of the season, we're able to monitor progress of our fish from the lower tag station shown here near the mouth of the river, all the way up into these different tributaries. So I don't have time to really dig in on some of the results, but this is just one summary slide to give you an indication of what we're finding. But basically, for both, I'm showing fat content here. This is fat measured by that fat meter I showed a picture of in the past slide. But we're also seeing very similar trends in size as well. But basically, what I'm showing here is the fat content of the fish in different groups in each year for each run timing group. So the columns here are the run timing groups, going from early, which is typically May, to middle, to the late run, to August. So that's kind of moving through the season. And then each row here in this plot represents a year from 2019 to 2021. And the colors indicate whether the fish were successful or not. So we divided each of the different tagging groups into fish that were successful and reached points upriver, and those that were not. And so what we find is, in general, particularly in the middle and late run, which is the largest component of the run, we find the fish that are higher that are larger, and fish that have higher fat content were more successful at reaching upstream points. So this, I think, has real implications in how we're interpreting past events, especially in years of relatively challenging migratory conditions and when fish are returning at small body sizes. We expect that relatively that mortality might have been elevated in those years. And then, of course, looking forward in time, if we're expecting warmer water conditions in the river or higher flows during the migratory period, we might find, and if the trend in size continues to decline, we're going to expect higher levels of en route mortality for these fish. So this is 3 years of data. We're hoping to continue in the future and to keep monitoring this. But this, I think, is a kind of concerning trend that we need to consider, especially as we're seeing all these changes occurring both in the oceans and in the rivers. So here's just a general summary of what we found through this migration study. General pattern is that early run fish have the greatest energy reserves. So the early run fish tend to go the farthest. Their spawning grounds are up at the headwaters of the river, and so they seem to have much higher energy provisioning compared to shorter distance migrants that generally arrive later in the season. By comparing the energy content of the fish in the lower river and the upper river, we conclude that about 50% of their total energy reserves are used. And of course, we're trying to understand how this might change from year to year based on the river conditions. But in general, about half of the energy that they start with is expended to reach the spawning grounds. Both size and energy content of the fish can explain migratory success. And so we're seeing generally the large fish and the fish that with higher energy provisions have greater migratory success compared to smaller fish and fish with at lower condition. And then just accelerated glacial melting can lead to higher flows that create a hydraulic barrier in some areas that haven't mentioned Wood Canyon, but this is a constricted reach in the lower river. In years of high flow, like what we saw in 2019, this could create a barrier for passage and make it much more difficult for fish to successfully complete their migration. Finally, something dramatic occurred in 2019. It's important to document that. I was involved in a paper that was published last year that documented over 100 cases of salmon die-offs during the summer of 2019. There was an intense heat wave that resulted in high river temperatures, low flows in many rivers, and increased incidence of wildfires. Each dot on this map represents a reported case of pre-spawn mortality of salmon, and the color indicates the species. The legend here is shown in the top right. And although not documented, we suspect there was increased mortality due to the high flows that I just described in the Copper River. But of course, it's very difficult to document that. We had a radiotelemetry project going at the time, but in all of these other cases, basically, if the water is clear and relatively shallow, you could see fish that died before spawning. But in a river like the Copper River, it's very difficult to detect fish that died prematurely. Okay, in the last couple minutes I have left here, I just want to describe some of the work I've done in Hokkaido over the years. I've been really interested in this species, Parahucho perrii, known as Ito in Japan, or Sakhalin taimen. I first traveled to Hokkaido in 2006, and I've tried to return almost every year. These fish are spawn in the spring, so I try to schedule my visits for the springtime. This is early picture of me releasing an Ito that I found stranded in a tributary in the Sarafutsu River. I've worked with a number of Japanese colleagues over the years, and definitely want to acknowledge Dr. Michio Fukushima, who's been a close colleague of mine, works for the National Institute of Environmental Studies in Tsukuba. He introduced me to these fish, and ever since then, I've been really fascinated and really enjoy working with Japanese colleagues and working in these rivers in particular. I was part of a team that created a large protected area for these fish. We worked closely with OG Paper Company, and were able to protect over 2,000 hectares of critical riparian and floodplain habitat. So if you see the map on the left here, the yellow indicates areas that are under permanent protection. And this is an agreement that was produced with OG Paper and Wild Salmon Center. I was working for the Wild Salmon Center at the time. So it's a great example of conservation groups, the local Sarafutsu Ito no Kai, and a large company, OG Paper, working together to protect habitat for this endangered species. This is a picture on the right of a spawning pair. Of course, male is brilliant red color on the left, and a female on the right, spawning in a tributary of the Sarafutsu River. It's been really gratifying to work on this, doing the research, but also working toward conserving this important species. Part of the work in the Sarafutsu has involved estimating the number of adults ascending the river to spawn every year in a major tributary. This is a picture of the field crew from one of the years, and we're using a sonar called Eris. Ditson is an earlier version of this, but it's a multi-beam sonar system that creates a video image of fish. And in this case, we were counting adult Ito moving upriver. So we were able to monitor for 5 years, get estimates of the size of the spawning population. And we're hoping to do this again this spring with the help of Michio and other partners. I'm using my volunteer position at IUCN to help broadcast messages to a global community. And so one example of this is this fantastic freshwater publication that was published by Shoal, which is an NGO, environmental NGO, and IUCN and others. This came out in 2022, and it was great to have Ito highlighted in this. Even on the cover, you'll see a picture of Ito on the cover of the publication. So we're really hoping that this raises global awareness about the species and the importance of protecting them and protecting their habitat. And I talked about the importance of climate change and heat waves, and basically this happened last year, and the year before last in 2021 in August. Very intense heat wave in Hokkaido in August that raised water temperatures to a level lethal for salmonids and Ito in particular. And our partners were able to identify a significant mortality event of fish in the summer there, and it made local newspapers. I'm showing you a newspaper article from the Hokkaido newspaper on the right side here. And so it's got a lot of us concerned, and we're planning a workshop and a meeting this coming spring in Tokyo and in Hokkaido to discuss this and come up with a conservation strategy to try to protect these fish into the future. And finally, I just think it's important to highlight the Shiratoko World Heritage Site. Some of you in the audience, I think, are aware of this and involved in this. I was involved in reviewing the original proposal to include the site as a World Heritage Site back in 2005, and I've made several trips to the site over the years. There's been certainly positive changes that have occurred. Some dams have been removed or modified, but there's still plenty to do to restore streams to their natural state. I look forward to continue working with Japanese partners and with IUCN to conserve salmon and rivers in this really important region in Japan. That's all I've got. I appreciate your time. Again, thank you for Sapporo Wild Salmon Project for inviting me. And I also want to acknowledge Kentaro Morita and Hitoshi Araki for originally contacting me and helping me with this, including the subtitles and translating this presentation for this special event today. So hopefully I'm available today virtually to answer any questions, but I appreciate your time and have a good day.